Very good afternoon to all the EAPCET aspirants. So here we are in the chapter Sexual Reproduction of Flowering Plants, which is a very major chapter. Every year you will get at least 2 questions from this chapter in your EAPCET exam. Let's get started. Ma. So here from this chapter, what are the concepts you have to deal with and how you have to learn it. So look at it first. Sexual reproduction of flowering plants, the very first thing is you have to fragment this chapter into 3 domains, into 3 domains which include pre-fertilization events, fertilization events and post-fertilization events. Okay, pre-fertilization events, fertilization events, and post-fertilization events. So here we go with first pre-fertilization events. First we will go on with pre-fertilization events. In pre-fertilization events, we will come across the male reproductive structure and female reproductive structure and the formation of male gametophyte and female gametophyte. Okay, that is microsporogenesis, formation of microspores and formation of megaspores under megasporogenesis. So here under pre-fertilization events, what are the contents to be covered? Structure of male and female reproductive structures of a flower, and then microsporogenesis followed by megasporogenesis. Okay, microsporogenesis followed by megasporogenesis. Just a second. Megasporogenesis. Fine, right. After this you will be learning what? Pollination, pollen pistil interaction, double fertilization, triple fusion. Double fertilization or triple fusion is one and the same. And post fertilization events, we will be going through 4 post fertilization events which include seed formation. First is embryo formation, next endosperm. First endosperm formation will occur and then embryo formation will start, after which the seed and fruit formation will happen simultaneously. Seed and fruit formation occurs simultaneously. These are pre-fertilization, fertilization and post-fertilization events. So here if you see, and then which is followed by, first, what is the reproductive organ of the flower? What is the reproductive organ of an angiosperm? Sorry, okay. Reproductive organ of an angiosperm is what? Flower. Reproductive organ of angiosperm is flower. Fine, right. This flower will be having 2 essential whorls and 2 non-essential whorls. Non-essential whorls and 2 essential whorls. Two non-essential whorls include calyx and corolla. Calyx consists of sepals, corolla consists of petals. essential words. Essential words include andrecium and gynoecium. These are male and female reproductive structures. Andrecium and pistil or gynoecium. Pistil or gynoecium. Andrecium is also addressed as what? Andrecium is also, andrecium consists of stamens. Gynoecium or pistil consists of carpels. It can be single carpel unit or multiple carpel units. Fine, right? This is the basic description of a flower. Good afternoon, ma. Just continue with the class. If you are not understanding anywhere or if you need any support, you just drop a message in chat box. I will be addressing your doubts. Okay. Very good afternoon, Shweta. Continue with the class, ma. So, here if you see the reproductive structure of the angiosperm, which is flower, non-essential whorls and essential whorls. Non-essential whorls consists of calyx, corolla. Essential consists of male reproductive structure andrecium, female reproductive structure gynoecium. Andrecium consists of stamens and gynoecium consists of pistil or carpels. Excuse. Where are these essential and non-essential whorls present? They are present over the receptacle or thalamus. Okay, they are present over receptacle or thalamus, which is over the pedicel. Okay, which is present over the pedicel. This is the basic description of a flower. Now we will go on understanding about how the flower is formed in a flowering plant. So, the decision of making a flower is taken far prior to the flower formation. What happens in the beginning is the shoot apical meristem over here. Okay, shoot apical meristem will turn to floral meristem first. Okay, and then floral meristem will develop an inflorescence axis. Inflorescence axis will form a floral bud. Later, floral bud will develop into what? A flower. Okay. So, here if you see, first is what? Shoot apical meristem. Shoot apical meristem will turn to what? Floral meristem. Floral meristem. How shoot apical meristem will turn to floral meristem? By reducing the internodal length. Okay. By reducing internodal length. Fine, right. This floral meristem will mature, or this will form into what axis? Inflorescence axis. Inflorescence axis. Inflorescence axis will form a floral bud, or will first form a floral primordia. Floral primordia will later develop into a floral bud. Floral bud turns to a complete flower. Clear, right? This is how a flower is formed, and this sequence you have to remember, very important, because we can expect a statement based question from this area. Assertion, reason or statement based we can expect from this area. So, first shoot apical meristem followed by floral meristem and then inflorescence axis, floral primordia, floral bud, which gives rise to what? A flower. This is the sequence of change for flower formation. Clear, right, ma? Yeah. Next you see a stamen. Okay, andrecium consists of what? Stamens. Fine, right? Each stamen has a bilobed anther and a long filament. So, here you can see male reproductive structure andrecium and female reproductive structure, that is gynoecium, present on the same flower. So, this flower is addressed as what? Bisexual flower or monoecious flower. Now, here you see, so each stamen will consist of what, ma? Each stamen will consist of a long filament, okay, a long filament and a bilobed anther. The filament proximal end is connected to, proximal end of the filament is connected to thalamus or receptacle, thalamus or receptacle, okay, and the distal end of the filament is connected to bilobed anther, bilobed, dithecous or tetrasporangiate anther, tetrasporangiate. What are the meanings of these terms? I will let you know, but then it is very important to remember, okay. So, each stamen will consist of what, beta? A long filament thread like structure called as filament. This proximal end of the filament is connected to thalamus or receptacle and distal end of the filament is connected to anther. Most of the anther in angiosperms are bilobed, dithecous and tetrasporangiate. Clear, right? So, here you can see filament whose distal end is connected to the anther. Here you can see the clear structure of anther, ma. Anther is bilobed. This is known as lobe 1 and lobe 2, okay. Lobe 1 and lobe 2. Can you see here? This is one lobe and this is one lobe. Each lobe will have two theca. This is one theca, this is theca 1 you can say, and theca 2, theca 1 and theca 2. So, it is addressed as dithecous. In total bilobed anther, how many sporogenous tissues are there at the center? 4 sporogenous tissues, that is why it is known as tetrasporangiate. Now, you have understood now why we are addressing it as bilobed, dithecous and tetrasporangiate. Tetrasporangiate has 4 sporogenous tissue is present at the center. This is tetrasporangiate. Fine. So, here this is the diagram which is given in your text, where in the proximal end of the filament is attached to the thalamus and distal end is holding bilobed anther, and here you can see it has 2 lobes. These 2 lobes will later mature into pollen sacs, and the longitudinal line here will act as line of dehiscence at the time of pollen grain release. This is the structural description of anther and filament. Now, we have to learn elaborately the structure of microsporangium or anther, okay. Structure of microsporangium or anther, take down beta. Structure of microsporangium or anther. Here we go. Anther will have 4 wall layers. If you can see, the outer wall layer is epidermis. Next to epidermis, you have endothecium. Next to endothecium, you have middle layers. Next to middle layers, you have the innermost wall layer, that is tapetum. Fine, right. So, you see here first is epidermis. Epidermis is the outer wall layer. Here you can see epidermis. It is outer wall layer. It is made up of parenchymatous tissue. There are no intercellular spaces. It is uniseriate. It is single layered. Epidermis is single layered. Okay, and it also has some fibrous thickenings in archaeothobium. In archaeothobium, anther wall layer, that is epidermis, you can see some fibrous thickenings. Okay. So, here epidermis is parenchymatous, uniseriate, which is single layered, no intercellular spaces, for any of the wall layer of the microsporangium, and in arceuthobium you can see fibrous thickenings. Okay. Next is what? Endothecium. As it has fibrous thickenings in the epidermis, this is also addressed as exothecium. Fine, right? Next to the epidermis you have endothecium. Endothecium will have alpha cellulosic fibrous thickenings from the inner cellulosic inner tangential walls. Okay. So, endothecium from the inner tangential walls, alpha cellulosic fibrous thickenings will arise. Okay. So, from inner tangential walls, alpha cellulosic fibrous thickenings will arise. Fine, right? That is the next wall layer. These alpha cellulosic fibrous thickenings are hygroscopic in nature. Okay, they are hygroscopic in nature, which help in pollen grains release. Okay, they are hygroscopic in nature, which help in pollen dehiscence. Fine, right? Next is middle layers. Unlike epidermis and endothecium, middle layers are 2 to 3 layered. They are 2 to 3 layered. Fine, right? Middle layers are 2 to 3 layered, and they are ephemeral, which are short lived. As microspores inside mature to pollen grains, the middle layers will get degenerated and they will help in the pollen release. Okay, they are ephemeral or short lived. Fine. So, epidermis, endothecium and middle layers put together, they have a single unit of function, that is protection and dehiscence of pollen grains. Protection and dehiscence of pollen grains. Clear, right? Next is what, ma? Tapetum. If you see tapetum, this is the innermost wall layer. Here we will discuss the ploidy ones, because many a times you will get the question on the ploidy of tapetum. If you see the ploidy of an angiosperm, it has sporophytic plant body, so it is diploid. Flower is also diploid. So, male reproductive structure will also be diploid. Yes or no, ma? Now, anther also is diploid, filament is diploid, stamen is diploid. Yes or no? On whole, stamen is also diploid. So, here anther wall layers, epidermis is diploid, endothecium is diploid, middle layers are diploid, but tapetum is polyploid in condition. Okay, tapetum is polyploid. Fine, right? This is polyploid, and the major role of tapetum is to provide nourishment to the sporogenous tissues which are present at the center. To provide nourishment or nutrition to the sporogenous tissues which are present at the center. This will provide nourishment to sporogenous tissue which are present at center. Fine Noma. So, tapetum will provide nourishment to the sporogenous tissue which is present at the center. This is about tapetum. Fine, right? Now, this tapetum will also help in the synthesis of sporopollenin, which is the outer wall layer of pollen grains. Tapetum will also produce Ubish bodies. These Ubish bodies will serve mainly for the purpose of synthesis of sporopollenin and nourishment to the microspores. Clear, right? So, here tapetum will help in the synthesis of Ubish bodies and it will build up the sporopollenin, which is the outermost wall layer of the pollen grain. Okay. So, here the sporogenous tissue which is present at the center will contain microspore mother cells which are diploid. This is the complete structural description of microsporangium. To go in more depth, see beta, all the first year students, the structure of microsporangium is important for your EAPCET, for your IPE as well and for your NEET as well. If you can describe each wall layer, that is epidermis, endothecium, middle layers and then tapetum and a small brief description about sporogenous tissue, then in a wholesome preparation this concept will give you 3 benefits. Okay, wholesome preparation for NEET, for EAPCET, for your IPE. Okay, and then here we go with microsporogenesis. So, at the center of each sporogenous tissue, you have microspore mother cells. Okay, sporogenous tissues will consist of microspore mother cells. Microspore mother cells are diploid. Okay, this undergoes reductional division or meiotic division forming microspores which are haploid. Fine, right? So, microsporogenesis, the cell which is going to undergo meiosis is addressed as meiocyte or microsporocyte as well. So, microspore mother cell is also known as meiocyte or microsporocyte. Fine, right? So, this microsporocyte will undergo first meiotic division. After meiosis 1, 2 daughter cells are formed. These 2 daughter cells are haploid, and after meiosis 2, 4 daughter cells are formed which are haploid. These are haploid microspores. These are haploid microspores. These microspores will be in what condition? Tetrad condition. Fine, right ma? So, here microsporogenesis, where microspore mother cells will undergo reductional division forming 4 haploid microspores. Microspore mother cell, as it is undergoing the meiotic division, it is also addressed as meiocyte or microsporocyte. And remember one thing, each microspore mother cell which is present inside the sporogenous tissue will have callose wall over it. It is encoded by the callose wall. Once after the complete meiotic division, when microspores are formed, the daughter cells of meiosis are also known as meiospores. These are also known as meiospores or microspores which are haploid. These microspores, once after maturing to pollen grains, then the callose wall layer will be degenerated by the enzyme callase. Callose wall layer will be degenerated like this by the enzyme callase, and the pollen grains will be released. This will release what? Pollen grains. Pollen grains will be released. Fine, right now? So, here microsporogenesis, microspore mother cell, which is also known as microsporocyte or meiocyte, will start undergoing meiosis, forming 4 haploid microspores, and from the beginning itself callose wall layer is encoded with. After the formation of microspores also callose wall layer will be there, and after which the callose wall is degenerated by callase enzyme, pollen grains will be released. Most of the pollen grains, in 60% of angiosperms, they will be released in two-celled condition. Okay, pollen grains will be released in two-celled condition. Yeah, Ashra, practice questions will be there at the end of each chapter beta. At the end of each chapter, if you can go through the previous chapters, we have finished off many chapters from your first year. Telugu Academy might be in your NEET though, from all chapters I have covered. After each chapter, you will have EAPCET previous year questions practice. Okay. Yes, you will be having at the end of the chapter beta. Hold on, first learn the concept well, and at the end you will have EAPCET questionnaire practice. Okay? Yeah. If you want to go with the previous chapter, you can watch yesterday's class of modes of reproduction, wherein the questions are already dealt, EAPCET previous year questions. Once after the chapter of sexual reproduction of flowering plants is done, then we will go with the questionnaire practice of this. Fine, right? So here you see most of the once after microspores are formed, this microspore will be matured to pollen grains, and pollen grains will be released in mostly two-celled condition, right? So here if you see a microspore structure has a large vacuoles and one small nucleus filled inside what? Cytoplasm. And in cytoplasm you will have large vacuoles and a small nucleus. So this vacuole will occupy most of the space of the cell, and the nucleus here will undergo mitotic division, which is without a spindle. So you can call it as amitosis, amitotic division without the formation of spindle fibers or asymmetric spindle fibers. As such, what is formed? Two cells are formed. Large vegetative cell consists of large vacuoles. Okay, vegetative cell consists of large vacuoles. Fine, right? Vegetative cell consists of large vacuoles and irregularly shaped nucleus. Large vacuole and irregularly shaped nucleus. Fine. And small generative cell is present inside the large vegetative cell. Generative cell is small, it is present inside the vegetative cell. Okay, small generative cell is present inside the vegetative cell, and generative cell will have spindle shaped nucleus. It has spindle shaped nucleus. Fine, right? This is the sequential description or the change from the microspore structure to the formation of asymmetric spindle and then two-celled condition. This is the complete structure of a pollen grain. Now you look into this beta. Yeah, pollen grain structure. If you see the pollen grain structure, first let us go from the outer layer to the inner layer. Pollen grain wall is composed of, pollen grain wall is composed of, grain wall is composed of sporoderm. Okay, it is composed of sporoderm. Fine, right? This sporoderm, which is pollen wall layer. This wall layer has two layers in it. Outer wall layer is exine. Outer wall layer is exine. Exine is composed of sporopollenin here. Okay, complete exine wall layer is composed of sporopollenin. Fine, right, ma'am. So here exine will have few small pore-like structures which are known as apertures or germ pores. Okay, these are known as germ pores or apertures. Only at this germ pore region sporopollenin is absent. Why sporopollenin is absent in the germ pore region? You may get in the assertion reason type of question. Okay, sporopollenin is absent क्योंकि from that region of germ pore, pollen tube will arise whenever the pollen grain lands on the suitable stigma. Whenever pollen grain lands on the suitable stigma, from the germ pore region only pollen tube will start arising. And next is intine wall layer, inner intine wall layer. inner wall layer is intine. Intine consists of cellulose and pectin. Intine wall layer is composed of cellulose and pectin. Fine, right? So, here pollen grain wall is composed of sporodon, outer wall layer is exine. Exine consists of sporopollenin. I will tell you the importance of sporopollenin. Many a times we got question on sporopollenin itself. On the exine, there are small pore or aperture like structures called as germ pores, where sporopollenin is absent. Next, it is followed by intine. Intine is composed of cellulose and pectin. Here, if you see, in the inner cytoplasm consists of two cells, vegetative cell and generative cell. Just now we have learnt clearly about vegetative cell and generative cell. Vegetative cell consists of large vacuole and irregularly shaped nucleus. Generative cell consists of what beta? Small spindle shaped nucleus, and generative cell floats inside the vegetative cell. Here you can see generative cell is floating inside the large vegetative cell. This is the structural description of pollen grain, and now you see what is the major importance of sporopollenin. Sporopollenin is the most resistant organic compound. Okay, this is one of the most resistant organic materials or compound. This can withstand high temperature, high acidic conditions and high alkaline conditions as well. No enzyme is so far recorded which can break sporopollenin. That structure, that major toughest structure sporopollenin has. Okay, so sporopollenin is the major reason for palynology, where we can study pollen grains, which is majorly of taxonomic significance, and this place, because of the sporopollenin which is present in the outer wall layer, this is also important for the study of fossilization. Okay, this is important for the study of fossilization. Fine. And then taxonomic significance, it also has what? Taxonomic significance. Fine, right? Okay, this is about sporopollenin. And then here we have to go through few concepts from pollen grains which are very important. First is pollen viability. Pollen viability. Second will be pollen allergy. Followed by pollen products. And pollen banks. These are the four concepts to be addressed here after the structure of pollen grains. Okay, so here what is pollen viability beta? Pollen grains, once after their release, okay, the capacity or time period of the pollen grains to retain on the stigma, okay, to retain the compatibility on the stigma is addressed as pollen viability. Pollen grains, once after their release, the retaining capacity of the pollen grains once to reach the stigma, to reach the compatibility of the stigma is addressed as pollen grains, okay, is addressed as pollen viability. Ma'am, can you please explain the taxonomical significance part again? Of course beta, sure. See, sporopollenin is the toughest organic compound, right? It is the toughest organic compound where no temperature or acidic conditions or alkali conditions and no enzyme is so far recorded which can degrade the structure of sporopollenin. That important structure it is, right ma? So, this sporopollenin which is present in the exine wall will help the pollen grain to store for a major period of time, okay, and pollen grain structure will not be disabled, okay. As such, we can use the pollen grains which are of many different eras in the previous generations for the taxonomic study, okay. For taxonomic study, pollen grains which are stored for very long duration of time can be used as the reference, okay, for taxonomic study. That is how, because of the structure of sporopollenin, pollen grains have what? Taxonomic significance. Now you have understood this, ma? Now did you understood this beta? Let me know. If you are still facing any difficulty, you can ask me, ma. Yeah. Now, next concept is pollen viability. Pollen viability is the capacity of the pollen grain to retain its compatibility on the stigma once after its release, right? So, pollen viability is variable for variable pollen grains. Okay, good, ma. So, here pollen viability is variable for different pollen grains. Okay, for different plants it is variable. So, here if you see, just 30 minutes of pollen viability will be there, sorry, just 30 minutes of pollen viability, 30 minutes. Once after the pollen release, it can fall on the stigma and germinate only within 30 minutes. That is observed in cereals like rice and wheat. In cereals like rice and wheat. And pollen grains will retain their viability for months. For months you can see in Rosaceae, Leguminosae and Solanaceae. Rosaceae, Leguminosae and Solanaceae. Fine, right? You can remember this as a mnemonic like RLS or SLR. Fine, right? But remembering the examples is very important here. Next is pollen allergy. Most of the people will be sensitive for the pollen grains, okay, and this will trigger in the people who have some lung disorders, especially bronchial afflictions, okay. For example, if we can take the pollen grains which are released from Parthenium, which is carrot grass, and Amaranthus and Chenopodium. The pollen grains which are released from these plants, pollen will be allergic to most of the people, and this may also cause bronchial afflictions. Fine, right? This is about pollen allergy. After which you can go through what? Pollen products. Most of the pollen grains are nutritive. They are highly nutritive. So, these pollen grains are being used in Western countries as pollen tablets and pollen syrups. These can also be used as, these are being used as food supplements by athletes. Fine, right? So, this is about pollen products. Next is pollen banks, that is preservation of pollen grains. Okay, preservation of pollen grains. How you can preserve the pollen grains? Pollen grains can be preserved in -196 degrees Celsius in liquid nitrogen, which is addressed as cryopreservation. Fine, right? This is about pollen banks. Just now we have learnt no taxonomic significance. So, those pollen grains which are being stored for generations can be used for taxonomic studies. As such, it has taxonomic significance. So, these are the concepts to be highlighted beta. First is structure of microsporangium, that is anther, where 4 wall layers are to be highlighted and learnt in sequence, after which microsporogenesis, how pollen grain is formed from the microspore mother cell, after which structure of pollen grains, and then pollen viability, pollen allergy, pollen products and pollen banks. With this, the male reproductive structure will be completed. After this, we will be going through what? Female reproductive structure, that is gynoecium or pistil. So gynoecium or pistil can have single carpel or many carpel. If it is having single carpel, you will call it as what beta? Monocarpellary. Monocarpellary. If it is having multiple carpels, okay, multiple carpels, this is known as multicarpellary. Monocarpellary and multicarpellary. Here if you see, each carpel will have a single unit of stigma, style and ovary. Okay, it has single carpel, single unit of stigma, style and ovary. So stigma is the receptive site which is receiving what? Pollen grains. Then this long tube in which pollen tube will grow, this is style, and basal bulged portion is called as ovary, inside which ovules are present. Okay, inside which ovules are present. Clear, right? So stigma, style and ovary will form one carpel unit. So as we have discussed, based on the carpel units, you have, just a sec, based on the carpel units you have monocarpellary and multicarpellary. Here we go beta. First is gynoecium or pistil. It can be monocarpellary or multicarpellary. Single carpellary, monocarpellary examples include mango, coconut and pea. Multicarpellary, which has many carpel units. Many carpel units, they can be arranged as free or in attached to each other. If pollen grains, if pistils are free, okay, if pistils or carpels are free, this is addressed as apocarpous condition. Best examples include lotus, rose and michaelia. If pistils are fused, if pistils or carpels are fused, that is syncarpous. Carpels fused is syncarpous. Examples include mustard, tomato, hibiscus and papaver. This is syncarpous, which are fused, and here carpels are free, that is apocarpous. Clear, right? Clear till here? Monocarpellary examples mango, coconut, pea. Multicarpellary, the carpels can be arranged freely or they are fused. Carpels arranged freely include lotus, rose and michaelia. Syncarpous condition where carpels are fused includes mustard, tomato, hibiscus and papaver. Clear till here ma? Now at last we have to observe the structure of ovule which is present inside the ovary. Okay, ovary, which is the basal bulged portion. So once I will make it clear here, this is pistil and the basal bulged portion is ovary. So this ovary consists of what? Ovules. Let me arrange the ovules in the form of marginal placentation, where at the ventral suture of the ovary, ovules are arranged in the straight line. Okay. So each ovule, like how it will be, most of the angiosperms will have inverted ovule. Okay. Most of the angiosperms will have inverted ovule. Inverted ovule. So here you can see inverted ovule, and now we will see parts of the ovules. This inverted ovule is known as anatropous ovule. Anatropous ovule. Fine. Anatropous ovule, parts you observe. Parts of anatropous ovule or ovule. So ovule will have first stalk of the ovule is known as funicle, through which ovule is attached to the ovary. Just like how in the mother's womb the fetus is attached by the umbilical cord, right, where from the umbilical cord the fetus will get the required nutrients to grow and develop. In the same way, the ovule which is present in the ovary will have funicle with which it can get attached to the ovary, and this gets all nutrition from the ovary. Yes, you can learn in our platform. You can learn in our platform. In NEET World, you have both offline and online facility. You can get into this. Fine, right? If you want to go through, you can call on to the number. Okay, you can call on to the NEET World number which is in website. Here you see the funicle. Funicle, which is the stalk of the ovule. So, first is what? Funicle. Funicle is stalk of the ovule. Next, small pore like structure, okay, which is connecting funicle to the ovule, okay, small structure which is connecting funicle to the ovule is called as hilum. Second is what? Hilum. Hilum connects funicle to the ovule. Fine, right? The small pore which is present at the base of this ovule is known as micropylar end. Micropylar end, which is pore like structure in the ovule. Fine. And the basal bulged portion here, opposite to the micropylar end, you can see chalazal end. So, fourth is what beta? Chalazal end is opposite to micropylar end. Fine. And here inside the ovule you have cells. These cells are known as nucellus cells. Okay, nucellus cells. Mostly the ploidy of nucellus cells will be asked. The ploidy of nucellus cells is diploid. Okay, nucellus cells ploidy is diploid, which is present inside the ovule. Fine, right? So, this diploid nucellus, one of the nucellus here will get differentiated into megaspore mother cell, that we will look in the next class under megasporogenesis. So, as of now we are clear with the ovule structure, and in our next class we will look into the types of ovule, megasporogenesis, embryo sac structure, pollination, artificial hybridization and double fertilization. In our next class, we will be looking into all these concepts. So, here in the structure of ovule, if you see, most of the angiosperms will have anatropous ovule and it has a stalk, that is funicle, and hilum will connect the funicle to the ovule. Next is micropylar end, which is the pore present in the ovule, and chalazal end, which is opposite to the micropylar end, and lastly nucellus cells which are present inside the ovule. Fine, right? The ploidy of all of this is diploid and nucellus is also diploid. This is the complete structural description of the ovule. Fine, right ma? So, that is all for today's session. Most of the students, I would like to end the session by addressing students actually. Most of the students who are particularly in the 12th standard, at this phase you will be very stressful, I know, because your IPA exams are heading up and you are also focusing on NEET and few of them also on the EAPCET. My advice for all of you is beta, look into the concepts in detail. There are many concepts where you can learn it for 3 exams together, all together, okay. Don't just mug up the answers for IPA exam. Learn the concept in detail, learn the subject in detail, which so that you can attempt it both in the objective form and the subjective form. Okay, in that way you have to learn. Fine, right? And for IPA exam, so I suggest you to finish off the bulk concepts in detail. If you can see in our channel, you can go through all the long answer type of questions from Botany are already done. You can go through that, okay. So, this is my humble advice to all of the students. Don't just mug up your questions and answers for IPA exam. Go concept wise, okay. Go concept wise and learn the concepts in detail so that you can attempt it both for the objective and subjective exams, okay. So, yes, keep learning with joy, okay. Don't get stressed anywhere. Keep learning with joy and don't have high expectations on results. Enjoy this process of learning and this process will be fruitful at the end, okay. Yeah, thank you. Thank you, ma. Thank you.